everyone. I'm Elizabeth Schneider, Senior Global Marketing Manager here at NanoString, and welcome to today's webinar, sponsored by NanoString and presented through LabRoots. So we have the pleasure of having two presenters today. Firstly, we will have Vikram Devgan, who is Senior Director, Spatial Genomics Business Unit at NanoString Technologies. And he will introduce the CosMx Spatial Molecular Imager, spatial biology at single cell resolution. And he will be followed by Dr. Chen Zhao, who's an assistant clinical investigator, medical oncologist, immunologist, physician scientist at the Center for Cancer Research, National Cancer Institute at the NIH. And he will be presenting on interrogating tumor-immune interactions in AML patients receiving immune checkpoint inhibition treatment. So today, we encourage you to participate by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. So let me introduce Vik. So Dr. Devgan is a Senior Director of Spatial Genomics Business at NanoString Technologies, and he received his Ph.D. in molecular biology and conducted postdoctoral research at Harvard before moving to the biotechnology industry. He also received an MBA from the University of Wurzburg in Germany and has spent many years as a scientist developing new technologies and products for clinical and translational research. Thank you so much, Vik, for joining us. We're looking forward to your presentation.
Thanks, Elizabeth. Hello, everyone. Welcome to NanoString Spatial Biology webinar. Comprehensive understanding of spatial biology requires integration of information at three different resolutions: multicellular, single cell, and subcellular level. NanoString is dedicated to develop spatial solution for all three spatial resolutions. We already have Genomics DSP platform available in our market. Genomics DSP, with its whole transcriptome analysis and high throughput, has become a method of choice for multicellular spatial analysis. Earlier this year, we have launched our second spatial platform called CosMx Spatial Molecular Imager. CosMx enables spatial analysis at single cell and subcellular level. In my talk today, I'm going to focus majorly on CosMx SMI. So let's start with, what does CosMx SMI do? CosMx enables quantification and visualization of 1000-plex RNA, 100-plex protein, expression level at single cell and subcellular level in a morphologically intact tissue. You are looking at raw data coming out of CosMx instrument with melanoma tissue FFP section. Here, teal color lines represent well-defined cell boundaries of single cells. And these tiny dots are images of RNA transcripts. One dot is equal to a one RNA transcript. So you are getting a complete digital quantification. CosMx enables quantification of more than 1 billion transcripts across 500,000 cells in a one single run. And that is the power of CosMx. Now, we all are well aware of single-cell RNAseq technology. It's a good technology. At the end, it yields cell types as well as genes associated with different cell types. But what it cannot provide is the information about where these cell types are located and how they are interacting with each other. And that's where CosMx comes into the play. CosMx fills that gap and enable analysis of both. CosMx not only provide the cell types, but also the exact physical location of these cell types. Once you have spatially resolved gene expression map at a single cell resolution, you can start asking multiple different biological questions. But before jumping into the applications of CosMx, let's look at how CosMx SMI works. CosMx uses single molecules in situ hybridization chemistry. Both RNA as well as protein targets are identified by in situ hybridization of specific probes or antibodies linked to a DNA barcode, as shown here as a readout domains. And these readout domains are decoded by cyclic imaging of fluorescent tracked reporters. CosMx needs up to 5 probes per gene, and the target binding domain of these probes are very small, just 35 to 50 nucleotide long. And that is why CosMx works beautifully with small transcript as well as with FFP sample with relatively degraded RNA. Now, let me walk you through what CosMx does in a detail. So please keep in mind, everything is automated and you don't have to worry about any of these. But to introduce the concept, I have divided the whole process into 6 different steps. So you start with tissue section on a standard morphology, standard pathology slide. And after a very simple sample processing, you upload that slide onto an instrument. Whole process starts with a very rapid, low resolution scan of the whole slide to detect or locate where the tissue section is. And then the tissue section is overlaid by the morphology marker staining. CosMx has 5 optical channels. It uses 4 for protein-based morphology marker staining, including cell membrane staining, and 5th for DAPI-based nucleus staining. Upfront morphology marker staining provide a great flexibility for CosMx users. It enables researchers to do either analyze the whole tissue section or focus on a small biologically relevant tissue area based on the morphology marker stains and get a higher throughput. Once you have selected a tissue area, CosMx goes through a cyclic imaging process and generate a composite image of all transcript of targeted genes. Then, CosMx uses data-driven cell segmentation method and define accurate and precise single cell boundaries, as shown over here. Followed by that, CosMx allocate detected transcript to single cell with well-defined boundaries. And once you have this information in your hand, you can start building different biological stories. For example, you can use our cell typing and mapping algorithm and define a cell atlas of the whole tissue section, as shown here with a non-small cell lung cancer tissue FFP sample. So CosMx analyzed around 120,000 cells in 20 millimeter square tissue area and detected around 18 different cell types, including 4 subtypes of T cells. Power of CosMx is it is a true single cell in situ expression analysis system. So you can validate all your cell types by visualizing expression of cell type marker at a single cell level. And seeing is believing, right? So you can publish your result with very high confidence. Now imagine you have such a data from 1,000 genes, and you can select any gene or a set of genes, visualize their expression inside the whole tissue section at a single cell resolution. And then you can start asking different biological questions. As an example, this is a study done by Jake Estes' lab from OHSU. So they are interested in understanding effect of certain pharmaceutical drug on a SIV infection. They used CosMx to analyze treated and controlled lymph nodes of SIV-infected rhesus monkey. COSMIC detected 14 different cell types and 4 tissue microenvironment niches in a lymph node tissue. Further analysis indicated that these SIV-infected cells are majorly or predominantly located in a B cell follicle of control lymph nodes. And they are dramatically or drastically reduced in a treated lymph nodes, as they have shown here with a red colored dots. So red colored dots are SIV-infected cells. And in addition to that, COSMIC enabled researchers for the first time ever to distinguish between productively infected cells from the uninfected cells. And this has made possible because of the super resolution of COSMICs. COSMICs has 50 nanometer resolution on XY axis and around 200 nanometer resolution on Z axis. Because of such a high resolution, researchers can easily visualize SIV gene expression in the cytoplasm of productively infected cells, whereas in uninfected cells, this SIV gene expression is located or trapped in a plasma membrane. However, the power of COSMIC is its high plex, which enable researchers to not only do cell typing and neighborhood analysis, but also allow a deep dive into the biology. As researchers used the 1000-plex COSMIC panel to define SIV infection biomarkers based upon a spatial location. Additionally, COSMIC has very high sensitivity. It enables detection of low expressor, medium expressor, as well as high expressor. COSMIC has a very high dynamic range of around 5 logs and enable detection of 100 of transcript per cells. As shown here with a study done by Grant Kohler's lab from University of St. Louis. So they are interested in defining cell atlas of human islet of Langerhans. They analyzed more than 1 million cells across 9 different samples and detected more than 500 transcript per cells. COSMIC enabled detection of 22 different cell types in a pancreatic tissue of human. However, this project is focused on endocrine cells, and COSMIC has successfully detected pancreatic beta cells, alpha cells, as well as relatively rare delta cells. Researchers used COSMIC to define cell atlas of more than 400 islets of Langerhans. And the cellular composition of these islets varies from based upon a human population or a groups. In a normal group, islets are majorly consist of endocrine cells, and beta cells are the predominant cell population. Whereas in a type 2 group, type 2 diabetic group, these islets are infiltrated by other cell types, including mesenchymal, fibroblast, endothelial cells. And it's important to note that researchers are able to make this observation only because of the spatial analysis. As the dissociated single-cell RNAseq technology, with that technology, researcher would have lost that information. The most drastic difference was observed with metabolically healthy, obese human group. Here, islets are very large in size and predominantly consist of alpha cells. So far, I have talked about the RNA capability of COSMICs. However, RNA is not the whole world. Protein is equally important, if not more. And you need high-plex RNA analysis to define or to understand the comprehensive function of cells. But at the same time, you need a high-plex protein analysis to get the absolute architectural understanding of a tissue. And CosMx provide both high-plex RNA and high-plex protein analysis. CosMx has already demonstrated 104-plex protein analysis at a subcellular level. I would like you to concentrate on lower left-hand image. Here, HER2 expression is highlighted with a yellow color, and it is predominantly located on a plasma membrane. Whereas PanCK is highlighted here with a pink color and predominantly located in a cytoplasm, whereas nuclear is stained with DAPI in blue. So this image beautifully indicates subcellular resolution of CosMx. In addition to a multi-omic capability, CosMx also provide a tunable throughput. CosMx enable analysis of up to 1,000 targets across 300 millimeter square tissue area with a throughput of 2 to 20 slides per week. CosMx is designed to enable both discovery and translational research. You can use CosMx to analyze, let's say, up to 1,000 target with 100 millimeter square area and with 2 slides per week. And on the other hand, you can also select to use CosMx to analyze 100 targets with a more biologically focused area, let's say around 16 millimeter square, with up to 20 slides per week. So flexibility is with the researchers. The one thing to keep in our mind is CosMx throughput is inversely proportional to the plex and tissue scan area. Additionally, CosMx provide robust data with all different sample types, with FFP tissues, tissue microarrays, organoids, as well as fresh frozen tissues. We have recently finished analysis of the whole tissue section of mouse brain fresh frozen samples. Here is a sneak peek of the data. So CosMx detected around 46 distinct cell types in a mouse brain, including astrocytes, neurons, oligodendrocytes, glia, and so many more. And stay tuned to learn more about this data in coming days. So this is my last slide. In summary, CosMx provide highest plex, tunable throughut, FFP compatibility, multi-omic compatibility, but on the top of that, CosMx provide completely integrated, easy-to-use system. Its sample prep is very simple and easy. It's based upon a standard ISH protocol. There is no tissue cleaning required. There is no gel embedding required. There is no amplification required. You can do a very simple sample prep and upload that slide onto an instrument. And CosMx has an integrated high-resolution microscope. So you don't need any additional instrument to conduct your experiment. Above all, CosMx comes with a cloud-based scalable compute and storage solution. And it has all the data analysis modules integrated into a cloud-based solution so that you can get in a one-touch button all that data analysis from CosMx done. With that, I would like to thank for your time. Thank you.
Thanks so much, Vic. So introduce Dr. Chen Zhao. So Dr. Chen Zhao is a medical oncologist, immunologist, and physician scientist focusing on tumor immunology and immunotherapy. He specializes in developing novel immunotherapies and using advanced tissue imaging techniques and spatial transcriptomic analyses to understand the three-way interactions among tumor cells, the immune system, and the microbes in the tumor microenvironment over the course of cancer initiation, progression, and therapeutic response. And as I mentioned earlier, Dr. Zhao will be presenting on interrogating tumor-immune interactions in AML patients receiving immune checkpoint inhibition therapy. Thank you so much, Dr. Zhao. We're looking forward to your presentation.
Thank you, Elizabeth. Yeah. Hello, everyone. I'm Chen Zhao. I'm an assistant clinical investigator from NCI. So today, I would like to share our research on interrogating the tumor-immune interaction in AML patients receiving immune checkpoint inhibitors. So this is all about our research, should not be considered as any endorsement of any products. So as we know, AML, this slide is a summary of all different types of cancer and the cancer-related deaths over the years. As you can see, we have already achieved a tremendous improvement of the lung cancer treatment. The cancer-related deaths was significantly decreased. However, leukemia still stay a very lethal disease, and actually there is a minimal improvement. As we know, in the normal bone marrow, this is normal hematopoiesis, and the blood cells, the myeloid cells, the lymphocyte, they all generated from the hematopoietic stem cells, and then they go to the progenitors, the precursors. And when in the AML settings, and certain lineage will stop differentiation, and the cell population will significantly increase, then you will see a lot of AML blast. This is leukemia cells circulating in the patient's blood or in the bone marrows. So the treatment of AML has significantly improved over the years. However, the treatment response is still mainly depends on the patient's genetic abnormality, which mainly determined by the tumor intrinsic properties. However, we know that the bone marrow transplant, actually transplant donor's bone marrow to the recipient, can greatly improve the patient response, especially in those patients who achieved a complete response after the chemotherapy. They do much better than those who did not. And this is just a brief summary of different bone marrow transplant strategies. And in AML settings, we are mainly use allogeneic hematopoietic stem cell transplant. So this background actually tells us the tumor-immune interaction, especially when you bring in someone else, donor's hematopoietic cells, the graft versus the leukemia effect can significantly improve the survival. So based on this knowledge, these findings, Dr. Chris Horrigan from NIHLBI, NIH, actually developed this clinical protocol to treat AML patient with pembrolizumab, which is the immune checkpoint inhibitor targeting the PD-1 receptors, and hypomethylating agent decitabine, which is the traditional AML treatment reagent for refractory or relapsed AML. Here I'm presenting a brief summary of the trials. Patient will receive pembrolizumab on the day 1, and then patient will receive decitabine on the day 8 to 12 and 15 to 19 in cycle 1, 3, 5, 7. So basically, we are combining the checkpoint inhibitors with the hypomethylating agent. So this trial also has a very special design. It allows us to obtain the bone marrow biopsy at the baseline or after the checkpoint treatment or after the combination treatment. And the result of the initial analysis of trial has already been published earlier this year. And as we can see, there are several patients who achieve a complete response. So this is very exciting in this patient population, because in the past, the refractory or relapsed AML patients respond poorly to the treatment. So as I mentioned before, the trial gives us a unique opportunity to look into the tumor-immune interactions. What does that mean? Because we can do the bone marrow biopsy, just a brief introduction like about the bone marrow biopsy procedures. So we put the needles into the bone, and the operator will draw the aspirate, which contains the bone marrow stem cells, the leukemia cells, and later after aspiration, we will also obtain a biopsy, which is a piece of the bone that contains the bone marrows. So the difference between these two is that all the cells here in the aspirate, the cells are isolated, they are not in the intact environment. So this will be analyzed with a single cell analysis platform such as 10x, allow us to look into the transcriptomes, or nowadays with the CySeq, you can also look at the surface proteins. And on the other side, with the bone marrow biopsy, it's an intact tumor microenvironment. And this is showing you an example when you look at under the H&E staining, and this pink part is the bone part, and actually you can later on we will zoom in to look at the leukemia cells and the immune cells. This is a staining of CD34, which is a marker for the leukemia cells as well as for the hematopoietic stem cells. So in this study, most of the patients, all the patients we selected, they have CD35 expression on their leukemia cells. So this allows us to look into this tumor microenvironment. So the pink cells here, this CD34 positive cells, this is leukemia cells, and you can see they cluster together, and surrounding it is those are the cells with the blue nucleus. Those are the stroma cells. They can be immune cells or other progenitor cells. So as you can see here, these are leukemia cells that express CD34. And the interaction between the leukemia cells and this black arrow point cells probably is not as important as the leukemia cell with its neighboring cells. So in this scenario, the distance really matters, and we really need to understand how the surrounding cells interact with the leukemia cells. So this prompts us to our hypothesis that the leukemia cells actually interact more with the nearby cells, especially the T cells, and the immune checkpoint inhibitors will change the cell properties in the surrounding cells. So we really want to know what are the cells near the tumors. So sorry, yeah, near the tumors, and the whether they are different from the cells that are far away from the tumors. So we started with using the digital spatial profiler, the Genomics platform, as just a brief introduction. It's a probe-based hybridization platform, which actually allows the users to select the probes near the certain protein markers. So in this way, first we want to know how are we going to define the near and the far. So using the traditional IHD staining, we can identify those CD34 expressing leukemia cells, and we can also identify the surrounding cells. So we measure all the distance, and we see there is significant increase of the enrichment of cells near the within 20 micron from the leukemia cells. So in this initial study, we decide to define the nearby cells by a distance of 20 microns. Then we need to use the imaging-based pipeline to identify those cells. So this is exactly how we did the experiment. So on every single slide, we have 3 samples. One is from the same patient, one is at baseline, the other one is a post-immune checkpoint inhibitor, which after the pembrolizumab treatment on day 8. And then later on, we have another bone marrow samples. The top one, those are the post-hypomethylating agent, the decitabine treatment. And we stain the slide with the cyto13, which showed all the nuclears, CD34, the leukemia cells in this setting, and CD3 will be the T cells. This is just a zooming of the RIs we select. You can see the blue nuclears, CD34, leukemia cells, and the CD3, those yellow, those T cells. Some of you may notice there are this kind of like big strikes of red signals. Those are the blood vessels. So in the bone marrow, the blood vessel, they're also positive for CD34. So this gives us a very challenging task. So we have 19 RIs per slide, and in order to collect all the samples, we want to process them in 1 hour, 16 minutes. During this 16 minutes period of time, we need to remove the background, those noise, and as you can also see, we also need to remove those blood vessels. Those are also positive for CD34, and we need to segment the cells. At the beginning, we try to just use the distance, but as you can imagine, if you purely use a distance to draw a circle, you will absolutely cut a lot of cells into half, and this will contaminate your analysis. And during this process, you also need to identify the cell type, which one is the leukemia cells, which one is T cell, which are the other cells, and by the end, we also need to measure the distance. Yes. So this is a very challenging job. In order to do this, actually, we develop a AI-based algorithm using a neural network, which actually allow us to remove the background, remove the blood vessel, do the instance segmentation, and identify different cell types based on their surface markers, and then perform the clustering. As you can see here, this is the image that processed after the imaging analysis pipeline, and we can separate the T cells into the near and far, the magenta cells, and the far T cells. Those are the cells in green, and the leukemia cells is in white. And the other cells, those are the cells that do not express CD34 or CD3. They also are divided into near and far. So in general, this is our study design. We have the baseline, post-pembro, and post-diazepam. We have 3 time points, and we identify the cell types based on the surface marker expression. Leukemia cells positive for cyto13, positive for CD34, but negative for CD3. T cells, they are positive for CD3, and non-T cells, those are the stroma cells, that are positive for cyto13 only, negative for both CD34 and CD3. And then we also have the spatial location, which is the center, and then the nearby, the cells within 20 microns or the cells more than 20 microns. So this is a very large study. After the QC control, 6 patients pass the QC. We have different cell segments. And here's just a brief introduction about the result. So as we can see, there is a dynamic change of the leukemia cells after at a different time point. On the left figure here, on the left side is the leukemia cells after the checkpoint, and relatively on the right is at the baseline. You can see there are multiple genes was enriched, so it suggests the checkpoint work, and probably this effect you are seeing here. This is actually the checkpoint probably work on the T cells, which I will show you later on, and this T cell attacks the leukemia cells. And the middle figure is comparing the effect from the diazepam versus the effect from the pembrolizumab. And on the right side, you can directly compare the baseline, which is on the right side, versus the leukemia cells that after the combination therapy, which is on the left side. So the most exciting part actually from our study is it enables us to look into the T cells at the different locations. So here, this is the result of the T cell transcriptome change at a different time point and at different location. The upper row show you the T cells near the leukemia cells. The lower row show you the T cells that those are far away from the leukemia cells. So we will start from the upper left, which is the baseline comparison to the after the checkpoint. As you can see, multiple genes were enriched after the checkpoint in the T cell near the leukemia cells. Relatively, if you look at the plot under it, it's a T cell far away from the leukemia cells. So the T cell 20 micron away, you see way less genes that were enriched. So we are still in the process of analyze this data. And in the middle part, you can see after the hypomethylating agent, multiple genes actually the effect diminished. And if we put all the genes enriched after checkpoint into a pathway analysis, actually it made a lot of sense. You see multiple pathways involving the cytokines production, interferon alpha, and even the IL-18 that were enriched in this group of T cells after the checkpoint treatment. And here, this is the comparison of the different T cell locations at a different time point. On the left side, this is the comparison between the T cells that are far away, which is on the left side, versus the T cells near the leukemia cells. You can see they harbor some difference, but after the checkpoint treatment, which is the middle figure, you see there is significant enrich of multiple genes involved in the cytokine and the cytotoxicity activity enriched in the T cells near the leukemia cells. And when you compare to other cells at a different time point, you see way-less genes, which here is also such. This might be caused by the effect. Actually, the other cells it contain different cell compartment. To further look into this effect, actually we use the simultaneous obtained single cell RNA sequencing data, did the spatial deconvolution, and when we look into the T cell subset, actually we see there are huge variety among different patients, and there is also dynamic change after the checkpoint at different spatial locations. This is also true for other cell types. Like we also perform the spatial deconvolution into the other cells, and you can also see dramatic difference at after the checkpoint treatment, also at different spatial locations. So in summary, from our DSP study, when we divide the T cell to the near and far different group based on the spatial location, the 20 micron. So the T cell next to the leukemia cells, they have a very unique transcriptional profile, and the immune checkpoint inhibitor definitely significantly affect the T cell next to the leukemia cells. And those hypomycinating agents, the dacetabine, they may change the T cell property, and this also deserves further investigation. However, here, as Vic mentioned earlier, so we divide the cells just broadly into two big groups based on like hard cutoff, like 20 micron. Definitely, we want to zoom into the single cell spatial resolution to unbiasedly study the spatial relationship. So we use the Cosmic platform, as Vic mentioned before. This is a probe-based platform using the barcoding strategy, which allows us to look at the transcriptome level at a single cell spatial resolution. So we perform the study with the exactly same batch of samples, and we look at the 3 time points, look into the core, and for each core we have this field of view, and we can stain for the surface markers. And here the magenta, they're showing the enrich, actually just showing all the genes, and you can see different spots enriched in the leukemia genes cells and the T cells. In general, from each core we have around 50,000 cells, and we have a read counts varies between 100 to 200. And this spatial technology actually allow us to also unbiased class cells in the tumor microenvironment into different clusters. This is a different clustering algorithm, and we were able to annotate, identify the T cells, the leukemia cells, the myeloid cells. And we can see they harbor very dramatic different transcriptomes, and the cell numbers, they also varies. And we are in the process of connecting the different modality and the spatial information with our single cell data set. So in general, I think with the DSP platform, we were able to see the dramatic difference between the T cells after the before and after the checkpoint treatment. Now with this new platform, we were able to zoom in to look into the exactly single cell spatial resolution, how the T cells interact with the leukemia cells. And then I would like to thank my mentor, Ron Jomain, for this opportunity. And also I would like to thank my students, my team, that Abigail Wanroli, who actually performed the study. And I would like also to thank our clinical collaborator, Dr. Chris Horrigan and Gigi Gui from NHLBI. Thank you, everyone.
Thank you so much, Dr. Zhao. So we had a number of questions that came in through the Q&A window throughout the presentation. So we'll go ahead and start with one question for you, Dr. Zhao. Using the same samples, how did your Genomics DSP data guide your CosMx SMI pilot study?
Yeah, I think this is a good question. I think there are two aspects. So first, actually, as I can show you earlier, so we actually performed the DSP study on multiple patients, and there are only 6 patients passed the QC. So first, we only select the patient who passed the QC from DSP for our CosMx the SMI study. And the second, the DSP itself also provide us a guide to lead our analysis to focus on the T cell the transcriptome analysis. So I think these two major parts that our DSP study actually help our CosMx SMI study.
Excellent. Thank you so much. Another question that came in for Vic. Can you explain the value of imaging the whole tissue section with single cell resolution?
Yes, I mean, the whole tissue section, it's when you're analyzing the whole tissue section, it takes guesswork out of the experimentation. You are generating data in an unbiased manner. And then, during a data analysis step, you will have more flexibility to select whichever region or a tissue area you want. So you are not missing anything. You are basically analyzing everything in one shot itself.
Thank you for that, Vic. So we'll go back to Dr. Zhao. What do you presume the distinct cell clusters found through your CosMx SMI pilot study indicate is biologically occurring in the tumor microenvironment in AML?
Yes. So this is a good question. I think the several distinct clusters we found, especially the T cells, I think it suggests that even in the tumor microenvironment, T cells still form clusters. They do not go along. They talk to each other. There might be two possibilities that actually we are pursuing now. One is that the leukemia cells near that group class of T cells, they are very different. They harbor different antigens or they have different cytokine milieux. So this is one possibility. Second is that T cells, there might be this cytokine to cytokine talking effect. The T cells may talk to each other and enable the function of surrounding nearby non-specific T cells. Yeah.
All right. Thank you for that, Dr. Zhao. We have another question for Vic. Can you elaborate on some of the applications of CosMx SMI, specifically as it relates to the platform multi-omics capabilities?
Yeah. So actually, I mean, when we are talking about application of CosMx, it has very broad applications. So just think about everything what single-cell RNAseq can do. CosMx can do that. In addition, on top of that, it's going to do that, provide all that information with the spatial context. So think about all the cell atlasing work like discovery and map of cell types, their cell state characterization on a protein side, as well as for the RNA, like phenotyping of tissue microenvironment, understanding the neighborhood niches and how looking into a differential expression of cell types with a spatial context again, characterizing whole ligand receptor interactome, then I mean, even the when you're looking at the subcellular level, especially on the protein side, the subcellular level quantification of the protein is really play a very important role in different biological processes, and you can achieve that and get that with CosMx.
Excellent. Thank you so much. Another question for you, Dr. Zhao. Let's see. Can you provide additional details about the development of the neural networks using Genomics DSP and AI-assisted imaging analysis pipeline, and possibly how cell type masking on DSP assisted with this?
Yes, so we are like in the manuscript preparation for the algorithm. So, but in general, the idea is that the images was processed at different layers, and we also we use the pixel-based classification to remove some of the background and also the blood vessels, and then we use the like state-of-art the cell segmentation tool, and then the cell identity was defined based on the protein marker expression, and we obtained the raw image from a DSP platform, and then we process raw-based image analysis pipeline, and then we actually put the mask back to the DSP platform, which will enable us to collect those signals.
Excellent. Thank you so much. So another question for you, Vic. Is the CosMx SMI process non-destructive to the tissue sections? And then also, can you customize the probes, both for RNA and protein?
Yes. So I'll take that, the last question first. Yes, both RNA and protein customization is possible with the CosMx. So CosMx on RNA side is going to have 3 different flavors. One is we are going to have well predefined panels, catalog panels, which can buy directly. And on top of that, the second flavor will be, customers have a few gene of interest or on which they have been working on from quite some time. They can add on around 50 genes on the top of our 1000-plex RNA panel. So it's more like you have the well-defined panel, but on the top you are adding your custom genes on top of it. And the third flavor is you can define your completely de novo panel. You can come up with with your gene list, and what you have to do is just provide us that gene list, and we can we can define those probes and manufacturers and validate it and send that to researchers. So it's a complete. There are 3 different flavor, including complete customization, and for RNA side, and we will have the same thing on a protein side. We will have customization of proteins, specifically add-ons to our catalog protein panels available in a Q1 of 2023.
Excellent. Thank you so much. And then the first part of the question was, is the processing of the slides non-destructive?
Yeah. So it's a little bit about how we define non-destructive. We can analyze its RNA, and then on the top of that, we can analyze both RNA and protein on the same slide. And if you want to redo and use this slide to do that some kind of bulk analysis, you can scrap everything and do the bulk analysis, whether it's on our DNA side or RNA side, that can be done.
Okay, excellent. So it sounds like in that respect, it's very similar to the Genomics DSP platform. Excellent. So we'll close with a question for Dr. Zhao. What kind of insight does combining spatial profiling data with single-star RNA-seq data provide?
Yes, this is also another very good question. So we feel that like single cell data, definitely, I think the sequencing depth is much, much deeper. However, the major question we had in the past is like, we never know whether actually those are isolated T cells interact with the leukemia cells, the tumor cells. So I think this is like a major advantage. Actually, we developed an algorithm to combine the multi-modalities, this multi-omics strategy, to put them together and to enable that actually identify those T cells profile near the leukemia cells.
Thank you very much. So it looks like this is going to conclude the Q&A session. Vic and Chen, do you guys have any final comments for our audience?
Thanks a lot for your time, for attending this webinars. And if you have any further questions, feel free to send it to us through email, and we'll be happy to take those questions.
Thank you, everyone.
All right, excellent. Thanks everyone on the call for tuning in for this webcast. I'd also like to thank LabRoots for hosting us. And also, if there were any questions that you didn't have time to submit today, or if we didn't get to any in the Q&A, these questions will be forwarded on to the speakers via the contact information you provided at the time of registration. And this webcast will be available on demand. LabRoots will alert you via email when it's available for replay, and we encourage you to share the email with your colleagues who may have missed today's live event. Also, I would like to mention that if you're interested in learning more about CosMx SMI, and you'd like to kind of get a flavor of the AGBT experience, we are doing a pre-AGBT event called the Spatial Multiomics Symposium. You can go to the events section of our website to register for that event. It is going to be in person in Florida, in Orlando, but it will also be virtual as well, so you can watch it streamed live. So you'll hear from people at NanoString, such as our Chief Scientific Officer, Joe Beecham, as well as some of our customers, and you'll learn both about some CosMx SMI experiments as well as Genomics CSP. So thanks everyone for joining. Thank you speakers. Thank you, Vic and Chen.
Thanks, Elizabeth. Thanks, everyone.